maybe it's reasonable to start. We have 1 or 2 minutes, but maybe the time to start. So I'm going to introduce Professor Untitled from the Karolinska Institute in Sweden. She has been educated in Germany, Hanover, and got also an associate professorship in Braunschweig University. The matter of interest of her work is related to the film production by microbial, and this is what she will start to discuss with us. She has been associated to the ICCBS field 6 years, member of the European Academy of Microbiology, distinguished fellow of COMSTEC from 2 years, and I'm very pleased to be chair to present how to find the most optimal treatment strategy for biofilm forming microorganisms.
Thank you very much for this nice introduction, and I'm very honored to speak today here. I would like to express my extreme gratitude to Professor Iqbal Chowdhury for inviting me to this symposium in the honor of the 80th birthday of Professor Ataur Rahman. And in honor of this birthday, I will present mainly what I have actually performed with Pakistani researchers, either at the Karolinska Institute as my collaborators, or here at the ICCBS with Professor Chowdhury and his team. So what we know about biofilms is that biofilms are a clinical problem. They are refractory to antimicrobial treatment and to the actions of the immune system. And a major question in this field is how to find most optimal treatment strategies for biofilm forming organisms. But first, I would like to present my institute. This is a new aula that has been built some years ago, where a lot of lectures are performed and administration presented. And then we have also here a new research institute that is called the Biomedicum. In this research institute, five previous departments of the Karolinska Institute are located to conduct the research, including our department, the microbiology, tumor and cell biology department. So we have stated that often, but still I would like to present what the definition of biofilm formation to introduce the field. So biofilms are basically matrix enclosed bacterial populations adherent to each other and or to surfaces or interfaces. And this definition of biofilm formation is very broad. Already it's an association of only maybe 2 microbial cells can be considered a biofilm or the beginning of biofilm formation. However, this definition does not include the environment or host components as extracellular biofilm matrix, which also plays an important role in biofilm formation, especially also in environmental biofilms. But what makes biofilm infection so refractory? The National Institutes of Health have estimated that up to 80% of the human bacterial infections are biofilm associated. And why is that? Biofilm formation is characterized by the production of an extracellular biofilm matrix and substantial physiological and metabolic alterations. These metabolic and physiological alterations make biofilm-forming organisms tolerant against antimicrobial treatment and the actions of the immune system. And eventually, this leads to chronic infections, which also leads to post-adaptation of the organisms. And as a host adaptation, for example, key genes required for biofilm formation are upregulated or even more upregulated than previously on the initial infection. And also the development of small colony variants can occur. These are additional natural mutants adapted to host conditions. Those mutants are, for example, impaired in the biosynthesis and or in menacanone pathway. They can also be impaired in CO2 fixation, carbonic anhydrase, and in thymidine biosynthesis. The extensive antimicrobial treatment can also lead to the development of mutator strains, and that can lead either to dead-end organisms or eventually also to organisms that are evolved and maybe can colonize new ecological niches. But how did biofilm formation of microorganisms arise? Biofilms are also already still formed today. Also, the first traces of biofilm formation have been identified 3.5 billion years ago. But similar biofilms, cyanobacterial biofilms, called stromatolites, are formed today, for example, here at the coast of Australia. And they are formed, for example, in hot vents, in, for example, here in Yellowstone National Park, spreading widely over the entire landscape and building up even high mountains. So you can imagine that biofilm formations are not only the most ancient multicellular structures, but they are also the most abundant structures. And the stress conditions that environmental bacteria in biofilms experience are very severe and extrapolate maybe in the clinical environment. However, we can even go much further. We can maybe hypothesize that biofilm formation has been intimately coupled to the origin of life. At the beginning, with the inorganic molecules that lead to the formation of organic molecules, and subsequently to the first microbial cells. The first microbial cells might not have survived without the protection by an extracellular matrix. And this extracellular matrix might have been, for example, extracellular DNA or cellulose, which is one of the most robust extracellular matrix components, consisting of only one sugar, glucose, and that sugar is 1,4 beta connected. So we can imagine that the first the first metabolisms that occurs needed to be protected in these harsh conditions. And this protection can basically only be given by also a biofilm formation of these organisms. How can we demonstrate that this hypothesis is correct? We can have genetic evidence for the early origin of biofilm formation is the presence of biofilm gene products encoded by the deepest branching bacterial phyla. So as an example, cyclic di-GMP is an ubiquitous second messenger that is directing the bacterial lifestyle transition between motility and sessility in all bacteria where it has been investigated. We find GGDF domain proteins with predicted diagonal cyclase activity present in species from the deepest branching bacterial phyla, such as Dictyoclomata, Coprocervobacterota, Callicerica, and Bipolaricauloda. And here is a phylogenetic tree showing the GGDF domain diagonal cyclases from these deepest branching bacteria related to, so to say, modern bacterium, Salmonella typhimurium, and one can see that these catalytic domains already are quite diverse, indicating that we have a maybe very early diversification of these biofilm regulators with that extrapolates maybe to diverse biofilm environments in the natural harsh environment on the primordial Earth. Our biofilm model is here displayed here. This is Salmonella typhimurium displaying a so-called red, dry, and rough biofilm morphotype on this agar plate compared to a non-biofilm forming Salmonella typhimurium colony. This biofilm is not only a model system for animal, plant, and human associated biofilm formation, but it can also, and in addition for environmental biofilm formation, but it can be also due to its tight interactions of the cells that we see in this type of biofilm matrix. And it can also be considered as a model for early biofilm formation on the primordial Earth. And when we consider cellulose as an extracellular matrix component, which actually is present in these biofilms, we can imagine that maybe those ancient pathways have been preserved in these bacteria, surprisingly, until today. But they have been, of course, remodeled and refunctionalized, and also in interaction of those bacteria now with evolved higher organisms like the human host. And I will come back to that later. So what we have, I already mentioned that what we have observed early on is that this type of biofilm that we investigated expresses two types of extracellular matrix components as a major component. One of the components is the exopolysaccharide cellulose, and the other component is the amyloid fimbria, in this case, perlite fimbria. Cellulose is produced not only by Salmonella typhimurium and by Escherichia coli, it is also produced by bacteria from the deepest branching, from deep branching phyla, like acidic bacteria species produce cellulose. So this is a demonstrated ancient biofilm molecule which produces a very specific biofilm type compared to the biofilm type that is produced by the amyloid fimbria. Transposon mutagenesis is a very powerful tool in order to find, so to say, unknown components of specific phenotypes. And we identified cellulose production in Salmonella typhimurium initially by transposon mutagenesis. Here is the current working model for cellulose biosynthesis in Salmonella typhimurium. And I would like to point your attention to a cellulase BCSF, which is surprisingly part of almost all cellulose biosynthesis operons. The role of a cellulase in cellulose biosynthesis has been for a long time enigmatic, and people did not know what the cellulase, the function of the cellulose, which is also present in plant cellulose biosynthesis. And in this case, it seems to promote cellulose biosynthesis. In the case of Salmonella typhimurium, we know that the cellulase is present to reduce cellulose biosynthesis, but in other bacteria, it also promotes cellulose biosynthesis. So this small protein plays a diverse role in cellulose biosynthesis. It can also be present in the periplasm, or it can be secreted. It can, so to say, aid the production of cellulose, but it can also aid the degradation of the cellulose. And that is all possible when one looks at the catalytic activity and the enzyme kinetics. So we investigated the role of the cellulase in biofilm formation and in microbial host interactions. And what we could see, and that was done, this work was done mainly also by Irfan Ahmad, that was at that time a PhD student in my laboratory. When we delete the cellulase in biofilms of Salmonella typhimurium, we can see consistently with a role in the degradation of cellulose and upregulation of biofilm formation. However, when we then complement with a cellulase on a plasmid, we don't see the expected phenotype of downregulation of cellulose biosynthesis again. And even when we express a plasmid with a catalytic mutants in the cellulase, we again see a similar phenotype as with the wild type overexpression, indicating that in this context, where we also have amyloid fibrilla present, we have a very complex interaction of the cellulase with the biofilm phenotype and expression of cellulose. While when we have exclusively cellulose present, and this data I don't present, but you have to believe me, we have a clear picture of the cellulase reducing cellulose biosynthesis, and even upon complementation, we see the expected phenotype. And what has surprised us very much is that this cellulose biosynthesis, which is present in biofilms, has been refunctionalized, and it has also a dominant role in microbial host interactions. In microbial host interactions, it basically trades against virulence. So cellulose production is an antivirulence factor. And other groups have shown that cellulose is even produced inside immune cells. And inside immune cells, it might lead to persistence of the organism, but it restricts proliferation. So when we delete the cellulase in this case, we overexpress cellulose, and we see a dramatic reduction of the replication of the organisms in immune cells. And we also see, when we delete the cellulase, a dramatic reduction of the systemic virulence in organ colonization. However, when we then delete the cellulose synthase BCSA, in both cases, we restore replication, and also we restore systemic virulence. So demonstrating that cellulose production or the cellulose or the presence of cellulose synthase is a major antivirulence factor of Salmonella typhimurium. The major biofilm regulator of Salmonella typhimurium is a transcriptional regulator, and this transcriptional regulator is called CSGD. CSGD positively regulates a digranular cyclase, and that the digranular cyclase is required for post-translational activation of cellulose biosynthesis by cyclic di-GMP. So we have identified cyclic di-GMP as an ubiquitous second messenger in bacteria. And when we have high cyclic di-GMP level, we could show we promote sessility on the single cell level. And when we have low cyclic di-GMP, we promote motility on the single cell level. So cyclic di-GMP was identified by us as an ubiquitous second, not only as an ubiquitous second messenger, but also as a regulator of the sessility, motility, lifestyle switch on the single cell level. And equally, we and others could show that cyclic di-GMP is an important molecule to promote the transition between chronic infection and acute virulence. And acute virulence is, for example, acute virulence phenotypes are, for example, invasion or the stimulation of pro-inflammatory cytokines. Surprisingly, this transition again involves the biofilm activator CSGD and cellulose biosynthesis as an antivirulence factor. CSGD is delicately regulated, and that is a work from another Pakistani, also a PhD student at the Karolinska Institute. And he discovered then upon deletion that the PSPA-DSP redox system is involved in the regulation of biofilm formation. So upon deletion of this PSPA-DSP redox system, we see a dramatic upregulation of biofilm formation. So this system suppresses biofilm formation. This upregulation does occur on the post-transcriptional level because we don't see any change in the transcription and requires cyclic di-GMP turnover protein, the phosphodiesterase STM3615 to be exerted. So upon deletion of this phosphodiesterase, unconventionally, biofilm formation cannot be upregulated in the absence of the PSPA-DSP redox system. The molecular mechanisms of this delicate regulation still needs to be elucidated. Another unconventional feature that we found, and that is the work of another Pakistani PhD student, is that we found unconventional cyclic di-GMP turnover proteins, STM1697 and STM1344, to be evolved and not neither hydrolyzing cyclic di-GMP nor binding cyclic di-GMP. However, these two proteins are still involved in biofilm and motility phenotypes, but they have just the opposite effect as conventional ERL phosphodiesterases. So why is, and those proteins act additive, they additively repress motility, and they additively promote biofilm formation, and they are involved in the systemic virulence of the organism. I don't present this here, but these two proteins basically target the major major regulator of motility, and this major regulator of motility is also delicately regulating the flagella in case of systemic infection, as those are molecular pathogen-associated molecular patterns, and due to the target of attack not only of the innate but also of the adaptive immune system. And just another example of the intimate interaction between cyclic GMP signaling and cellulose biosynthesis in streptococci, which conventionally human pathogens have lost the cyclic GMP signaling system. We see that in other microspecies of the genus Streptococci, for example, in the clade Salivarius, Pyogenes, Henryi, and Suis, and Bovis, we see the introduction of a cyclic GMP signaling system on a horizontally transferred genomic island in association with cellulose biosynthesis. So we can expect that these organisms also trade virulence against maybe survival in the environment and produce as a cellulose-based biofilm at some point, maybe even upon infection. A work that we have performed in collaboration with Professor Chowdhury as biofilm formation is a major biofilms are refractory to antimicrobial treatment. It is of course important to identify antibiofilm compounds, and we have identified the macrolide clarithromycin as an antibiofilm compound, while the azithromycin, the macrolide azithromycin, did not show antibiofilm effect. We can follow the dissolution of biofilm formation upon subinhibitory concentration of clarithromycin. Here, subsequently, these bacterial aggregates are dissolved, and surprisingly, we see upon subinhibitory concentrations of clarithromycin, we see a very specific response of the organism. And this very specific response of the organism is not only showing the targets of clarithromycin upregulated, like specific genes for ribosomal proteins. However, it shows that clarithromycin induces a substantial physiological and metabolic alterations of the entire bacterial metabolism. And this includes that, for example, the oxidative phosphorylation is dramatically downregulated, but arginine metabolism is, for example, upregulated. Yes, I just want, as I have not so much time, I maybe skip this part, but I just want to mention that another postdoctoral researcher in my group has worked on biofilms of Candida parapsilosis. Those Candida infections are the fourth most common cause of bloodstream infections, and 90% of candidemia cases results from device-associated biofilm infections. Our important organism here is Candida parapsilosis. It's the second or third most commonly isolated species in Candida bloodstream infections, and in some countries it can be even number one. Importantly, one of the only virulence factors identified in this species is biofilm formation. What we have observed is, when we looked at a panel of isolates from bloodstream infections, is that some biofilms some isolates did not form biofilms. So we were wondering whether there are conditions other than the standard conditions where those isolates maybe are also forming biofilms. And we found, surprisingly, that addition of manganese to the medium dramatically enhances biofilm formation in these non-biofilm forming strains and also in low biofilm forming strains, while this addition of manganese to high biofilm forming strains had only a minor effect. When we look at that under the microscope, we can see that upon addition of 2 millimolar manganese, we see this dramatic enhancement of biofilm formation and also a differentiation of the cell morphology in these biofilms, which then produce hyphae, pseudo-hyphae, and subsequently also hyphae under some conditions. This is shown for the different strains, while here with the high biofilm forming strains, we only observe additional differentiation of the biofilm, but the biofilm in the biomass enhances only not substantially. A major regulator of biofilm formation of low biofilm forming strains is the biofilm regulator BCR1. And this biofilm regulator BCR1 can also partially mediate manganese-dependent biofilm formation. So as we can see here, with no manganese addition, we see that BCR1 regulates biofilm formation, but it regulates it throughout all magnesium, all manganese concentrations. We see a BCR1-dependent biofilm formation. On the other hand, we could observe that when we delete the biofilm regulator BCR1, we promote cell differentiation upon low biofilm formation. And this leads us to a regulatory pattern where BCR1 promotes biofilm formation, but it inhibits differentiation of biofilm formation, for example, hyphal formation. The molecular mechanisms again have to be sorted out still for this novel phenotype. So this work was conducted by a postdoctoral researcher, Sulman Shafiq, also from Pakistani origin. Since my time is over, I skip this last part, and I would just like to thank all my co-workers and collaborators. Without them, this work would have been possible. And in the context of this symposium, I would especially mention the contribution of Irfan Ahmad, Naim Anwar, Sulman Shafiq, and also Sharif Hasni that did excellent work in my laboratory. And that is, I'm very pleased to be here. Thank you very much. All the collaborators, of course, also from the University of Karachi and others.
Thank you very much. Before we go into discussion, one or two rapid questions. Yes.
So, it's the cellulose. These are especially the metrics for biofilms, right? Okay. So, like in eukaryotic cells, so a lot of eukaryotic cells also make monolayers, and they have like heavily glycosylated proteins as it is present in the matrix. So all these cellulose are linked to certain proteins, or they are just free cellulose.
In the biofilm that I presented, the cellulose is co-expressed with another extracellular matrix component that are amyloid fimbria. And they form a tight interaction with this amyloid fimbria. And that one can see already macroscopically, this tight interaction. However, under other circumstances, the cellulose can also be produced without this amyloid fimbria co-expression. And then you have a more flexible cellulose macromolecule, which might interact also with environmental factors and surfaces, basically.
Thank you. Another question? The last one. No? Thank you very much. I have a quick announcement by the organizing committee today.
तमाम तालिब इल्मों से गुजारिश है कि जिन्होंने वर्कशॉप में रजिस्ट्रेशन कराई है वो गोल्डन जुबली हॉल में तशरीफ ले जाएं।
Thank you. The next speaker is Professor Dr. Rafat Ali-Siddiqui. He is currently working as Professor and Director of the Nutrition Science and Chemistry in the University of State Virginia. He has been educated in Pakistan, then got his PhD in Australia National University in Canberra. He has published more than 150 scientific papers and written several review articles on that aspect. His interest is in the area of cancer, inflammation, cardiovascular disease, and I give him the floor for presentation of work, role of change and nutrition in precision nutrition created.
Very much, Chair, for your introduction. First of all, I'd like to thank the organizer and my dear friend and brother, Dr. Iqbal Chaudhary, for the invitation. And it's really an honor and a privilege to be here to celebrate Dr. Abdul Rahman's 80th birthday. I could not have missed that. So once and Wigmore said that the food we eat can be either a safest and most powerful form of medicine or the slowest form of poison. And I'm going to show you, how this is very much true. So food, basically, we know these are the nutrients, and nutrients, we know that, typical nutrients are carbohydrate, protein, fat, vitamins, minerals, and water, which are essential for our growth and maintenance. Most of our body is composed of as you can see here, for water, proteins, fat, a very small amount, which are the other substances such as carbohydrate or the metabolites, which are produced during the metabolism of all these carbohydrate, protein, and fats. The food we eat or the nutrients or food-derived bioactive molecules, they basically get absorbed and metabolized, and these molecules are used mostly for producing energy. But they also catalyze some of the reaction. They are ligands for various signal, receptor signaling receptors, and they are also components of various in cells, cellular components. But most importantly, some of the bioactive molecules present in food, they are very powerful regulators, and they can affect metabolism. So nutrients we eat. Basically, we have a metabolic heterogeneity. We probably eat similar kind of food, but we can digest it differently and metabolize it differently. People certainly on different food intake, they have different kind of metabolism, but even people eat similar kind of food, but their lifestyle or the microorganism present in their gut or the environment they are living in, or they can greatly influence how these nutrients are metabolized. Other than this, the biggest role in the metabolism of nutrients that plays is by genetics and our epigenetics. So what we have, we know that we have all, cell as chromosomes, and chromosomes is express various genes. We have all similar kind of chromosomes and similar genes. The only difference is that we have gene variants, and these gene variants encode proteins which have different amino acid sequence, which makes their function different. Sometimes, it's the same enzyme, but in some people it may be more active, in other people it will be not active at all. So this actually contributes in metabolic heterogeneity. So as far as the nutrients are concerned, what we have, the food we eat, actually is a very powerful regulator of gene expression. So what we get is nutrigenomics, is the genes which are affected by the nutrients we eat. And what happens that because of the food we eating, the genes which are expressed or the environment we live, we have certain mutations. So we have different kind of genetic variations. Same gene, but different mutated forms, which actually contribute how the food we eat that get metabolized. So we have nutrigenetics is basically the variation in genes which helps or which affect the nutrient metabolism, whereas the food which helps into or which regulates the expression of genes. So they are interconnected. Just give you an example, like so nutrigenomes are like the metabolites we get either from diet directly or from our gut bacteria. They modify the food, and the product we get, the metabolites, they regulate epigenomes. Epigenomes is just simply the modification of histones and DNA methylation. So because of these two processes, certain genes are expressed or certain genes are suppressed. So this contributes overall in the nutrigenomics. But the genes that are expressed, they maybe varies from one person to other person, and that contribute to nutrigenetics. So just to give you an example, normally we have two sets of alleles, and they are usually homozygous. So we have the same kind of genes in both alleles. By mutation, one can change, and we have like the heterozygous. Still, they are normal, but if for some reason the both alleles are different than the original one, then we have a mutated gene. And all these mutated genes are arrived. It depends, how people migrate from one place to other place in different environment. So we have like ancestry, like Western Europe, European ancestry, Euro-Asian ancestry. And then that's how we have variation in genes. So typically, each of us has about more than 50,000 common inherited spelling difference in our genes for single nucleotide polymorphism. So it's the same gene, so only one nucleotide is different. And that nucleotide, for example, here GC is changed to C instead of GG, and it can have a profound effect how we digest our food. Now we have the ability is only possible because of the technology, and we can now measure about 2.5 million of these single nucleotide polymorphisms in a DNA sample using gene chip technology. So the food we eat, it has to go through the metabolic pathways. We have different metabolic pathways for protein, carbohydrates, lipids, and all that. So they break down into small molecules. So what happens because of the variation in genes, we get roadblocks. So some of the genes are inhibited or not active, so that results in the accumulation of certain metabolites or deficiency of certain metabolites. So what the situation we get that in the normal conditions, we're getting generation of metabolites certain amount in certain quantities. But if we have a roadblock here, like because of this single S and P, we get less amount of metabolites. And as I said, that some of these metabolites, they are powerful regulator of gene expression. So if you get less, in order to have a normal health, we need to supply these metabolites in diet to acquire the same concentration that what body needed to operate normally. So what we have done actually, this concept of precision nutrition came after precision medicine, and the lessons we learned from precision medicine. So typically, what we used to do is that we typical clinical trial, we have a population, give them some kind of drug, and then measure the effect. So what happens is that we get variable effects. Some respond good, some respond not so good, and some have some adverse side effect. But what happens is that because this is all mixed population, so at the end we get the data that this drug is not effective because it's all normalized. We have a big standard error, and the end data is okay. This drug is not useful. So what happens that if we can select our people, so if we do pharmacogenetics before and select our people who can respond better, or then we can adjust the drug to people who we think that they can respond better if they adjust the drug, and then we can select our people who might have the adverse side effect, then we can basically study the drug and target the drug only to those people which can benefit from that drug. So a drug which previously known as like useless, but by looking at the genetic makeup, it can be very useful. So what it is that like, so if we have no variation, person can have a standard dose. If we have genetic variation, maybe dose need to be adjusted. Or if we identify that this is too high, so dose the dose can be lower. So that's what happens that when we need to utilize nutrients, we get all kind of metabolic heterogeneity, and especially the gene polymorphism or SNPs, they cause either reduced or increased health risk, or because of this polymorphism, we can reduce availability of certain active metabolites. In some cases, we get improved responses, and some cases we develop tolerance. I'll give you some examples. There are plenty of examples in each case, but because of the time constant, I just limit it to give a few examples. So the SNP which are linked to reduce or increase health risks is the example is caffeine. We know most of us like caffeine, and the caffeine is metabolized in our body. The enzyme which metabolizes caffeine is P451A2, cytochrome P451A2, and the activity of the enzyme is actually greatly influenced by various environmental factors as well as by SNP. So what happens? Caffeine, the average metabolic rate of caffeine is half-life is about 5 to 6 hours. People have caffeine in 5 to 6 hours, half of it is gone. We don't see the effect of caffeine. But actually, caffeine half-life varies from 3 hours to 11 hours. Some people are, slow metabolizer of caffeine. Some people are fast metabolizer of caffeine. And this is because of the presence of this gene CYP1A21. So people with genotype AA, they are the fast metabolizer, whereas people with C SNP or CC, they are slow metabolizer. So what happens that this enzyme actually converts caffeine into certain metabolites, which are the activators of various metabolic pathways. So people who are fast metabolizer, they actually, when they have drink caffeine, it actually increases their endurance. This provides some beneficial effect, some kind of cardioprotective effect. They have lower risk of hypertension and MR. Whereas in case of people who are slow metabolizer, they have a variant here, actually it hurts them. It decreases their endurance and actually weakens worsening. Not only that, they are at increased risk of hypertension and myocardial infarction. So the fast metabolizer, when they drink at least 2 cups of coffee in a day, they are about 22% less likely to have a heart attack. Whereas the people who are slow metabolizer, if they drink 2 cups of coffee in a day, they are at an increased risk of 36% to have a heart attack. And if they drink 4, their risk get double. Now, the second example is the SNP linked to improved responses. Here is this enzyme catechol or methyltransferase or COMET. This enzyme involved in methylation or inactivation of some of the catecholamines like dopamine, norepinephrine, or epinephrine. What happens that there was a study done, and they found that when they give vitamin E to women, and it was like a 10-year study, they have a very mixed result. They did not find effect of vitamin E. But later on, when it become like known about this enzyme, so they look for this SNP and they select out the people. So what they found, that people with this SNP in this region, where this AA, the methionine is replaced by valine, actually they have about 50% less chance of developing cardiovascular disease, and they have reduced risk of developing colon cancer. So here's the SNP in the gene, and here's the data. So here's the placebo, and there's the vitamin E treated, this red one. So you see the SNP with methionine has a very beneficial effect of vitamin E, whereas methionine valine, this placebo here is black one, and then and the red one is actually slightly increased, but there's no significant difference. And then valine mutation is, again, there is no difference. So just having the proper genotype can be highly beneficial for this vitamin E treatment. Similar cases in colon cancer, that the colon cancer is reduced with this methionine genotype, whereas valine methionine there is no effect. And there's a data actually published, someone did the research here, is in the Pakistani population in various ethnic groups, like Sindhi, Punjabi, Balochi, and all that. And there's, so our population is predominantly methionine. So we are not at a risk of this enzyme activity. Okay, now the other one example is the SNP linked to reduced availability of active metabolites. And in this case, we know that choline and this is ethanolamine methyltransferase. There's an association with this because this enzyme involved in the synthesis of phosphatidylcholine. Choline is very important, but most of the people avoid using food containing choline because it is actually the food which has choline also has high cholesterol. So in US, most Americans do not actually achieve the adequate level of choline. And choline is very important, it's especially important for babies and the nursing mothers, because the choline is involved in neurotransmitter formation, it's involved in choline phospholipid formation, all the components in cell membrane. It's also a very powerful methyl donor, so it's methionine. The methionine is actually give methyls group for the methylation of histone and DNA methylation, which are powerful regulator of epigenetics. So in case of choline deficiency, what happens? Body has a system where this choline, the phosphatidylethanol is converted into phosphatidylcholine through this enzyme. But this enzyme is highly regulated by estrogen. So the estrogen concentration, which are typically present in premenopausal women and pregnant women, they are high enough to induce expression of these genes. But what happens that some people, they have polymorphism in this gene. In that case, this enzyme is not active and they cannot really convert phosphatidylethanol into the phosphatidylcholine. So the woman or the man with this genotype, especially CC, they are at a high risk of organ dysfunction. Men, in case of men, what happens is that because men have low level of estrogen, so they are at a higher risk at all the level if they have low concentration of or they have low intake of choline. So the dietary requirement, like older women, older men, is about like they are like 77% they are deficient in choline, like postmenopausal women 80%, younger women 46%, and so this polymorphism actually reduces the ability of estrogen, and they have an increased dietary choline requirement, and they are 25 times more likely to develop liver and muscle damage if they have a low choline diet. And these are some of the food sources where choline, by eating this food, you can increase the choline intake. So this is the distribution of this PEMT polymorphism in different populations. In Asia, it is not so common, but mostly is in Europe and in North America and in some African countries. So another example is polymorphism that is linked to non-tolerance. A very common example is lactose intolerance. We have this lactase which is present in our intestine. And what happens to lactase that we have a high expression in fetus. And as we grow older, the expression actually reduces. So it's in all mammals it happens except in humans. So, in some humans, what happens that they have persistence. So they still have high expression of lactase, but about 65% of humans worldwide they have this lactose intolerance. So what happens then? People who have lactose intolerance, lactose is actually not degraded, it enters into the large intestine where the gut bacteria acts on lactose and produce a lot of gas and acid, and you get bloating and discomfort and some kind of diarrhea. It's very uncomfortable. And is the distribution of lactose tolerance in different region. Indians, like in our population, people especially from Indian, in the southern region, they have about 60 to 70% lactose intolerance. And interestingly, it is not very common in European and American because of this gene polymorphism where they have this developed tolerance to the lactose. And this lactose intolerance is because of this regulator gene, MCM6 gene, which actually a switch, which actually regulates the expression of lactase. So in the intron, in the regulatory components, there is there's several actually polymorphism. So what happens that people who with C genotype, they cannot tolerate lactose at all. But if there's a mutation, they develop this lactose tolerance. And this historically, what happens that European people, they started using milk a long time ago as a diet, whereas the people in Asia and in Africa, they use animals mostly for meat, whereas in Europe they use animals for milk, and they develop the tolerance gradually. And this intolerance is because, as I said, there's a transition from T to C, and the C cytosine is heavily methylated. So it's an epigenetic mechanism. When it's methylated, this regulator will get inactivated, so it's no longer is going to act on and doing the lactase expression. Now, it's a few more minutes I will spend on epigenetics. Epigenetics is a mechanism where it regulates how the genes are expressed. It's mostly like histone modification, like methylation or acetylation phosphorylation, or DNA methylation. So what happens that the metabolites we take, they can be either gene suppressor or activator. So they induce a lot of methylation, which suppress the gene, or they can induce a lot of acetylation, which activates the gene. And our diet actually can be a very powerful factor which can regulate some of the genes and actually regulate how it influences the cancer formation. So some of the genes, the dietary polyphenol which are present, they inhibit the processes which involve with DNA methylation and histone methylation, DNA acetylation, whereas they inhibit the process where the histone get acetylated. So one example is folic acid. You know, the folic acid is very important, is a methyl donor, and folic acid, because histone modification or histone methylation, DNA methylation is very important, and folic acid contribute in the methylation process. Just to briefly, what happens that the DNA methylated on cytosine, like 5-methylcytosine, and methylated DNA is suppressed. But some of the dietary agents, what they do, they actually take reverse the process. So what they do, they take the methionine from S-adenosyl methionine and convert it S-adenosyl homocysteine. So basically, they're providing less substrate for this methylation process. So there are a number of compounds which do that, so they inhibit the methylation. And these are some of the compounds in our diet which can regulate genes because of inhibiting the DNA methylation process. And then histone modification, again, the histone get modified, is called acetylation or deacetylation, and we have a lot of nutrients which either do DNA methylation or histone modification, and then we have food which can actually do both methylation and DNA modification. Other briefly, the things are mRNA, which are also linked to diet. Diet also influences the biosynthesis of certain mRNA, which is involved in tumor oncogenesis or tumor suppression. And here's an example that some of the mRNA is linked to the nutrients we take, and they influence some of the organs we have. And then just for giving you an example, that cardiovascular system can be influenced by a number of miRNA, which are linked to the intake of various different nutrients. So again, in cancer, the number of nutrients they are linked, which kind of changes the expression of various miRNA, and that directly affects the progression of different cancer. For example, curcumin significantly changed the expression of 29 different miRNA in pancreatic cancer. EGCG present in T modulate expression of miRNA in hepatocellular carcinoma. MRI-16 is upregulated by ECGC and regulate the anti-apoptotic protein BCL2. EGCG treatment also induces apoptosis and down regulation of BCL2 in liver cell. Genistein, another compound which is present in soy products, also inhibit. It has also some anti-cancer properties in that first one in melanoma cell. So with this knowledge, we have like challenges, opportunity, and resources. To give you an example, since we know that people are because of this gene polymorphism cannot digest or eat certain foods, so there is opportunities for industries to develop new products. Now we have products like gluten-free. And gluten is also an example to lactose intolerance, which is also resulted with the gene polymorphism. Lactose intolerance, now you can find products which are lactose free and people can take them. Now it is very common, especially in USA, that there are a number of places where they test all this, can actually do the gene polymorphism and give you the data, and doctor can actually interpret and then advise you what food to avoid and what food to eat in order to get the maximum health benefits. And actually, I saw this paper we actually published from this institute, and I was very proud of saying this is a review article which is about different variants which are present in Pakistani population. So it's I encourage you to read this paper. It gives you, the idea that what kind of gene polymorphism present in Pakistani population. All we need to do, we need to basically knowing gene polymorphism can help find us precision nutrition for a group of population. We need better SNP information for Pakistani population for healthier life. And then we have opportunities here for developing nutrition formulas to target a special group. So I stop here. Thank you very much. And I apologize, I rushed into towards the end because of the time constraint. Thank you.
Thank you very much. It was easy, and I will open directly the floor for discussion.
Thank you so much for such a great presentation.
You're welcome.
So simple like that. How a common person can predict himself that he has fast metabolism or a slow metabolism for gene? As you categorize, there are the different effects for the fast metabolism and slow metabolism.
Yeah, it's very easy. If you take a cup of coffee, you cannot sleep at night. You're slow metabolizer. So, but no, yeah, if you want to prove, then you have to basically test and look for the polymorphism. But some of the things we can learn from our, how our body respond, from drinking tea or coffee. If you think that you cannot sleep, then you obviously you are slow metabolizer, so you want to avoid it. Your question?
Yeah. Thank you very much, Dr. Bhai. Very interesting and very useful lecture. I just wanted to know that this whole area of drug-food interaction, because people are taking medicine for chronic diseases, and then they go through all this episode of different kinds of interactions with drugs. How this drug-food interaction study and how relevant it is for the nutrition.
Yes, there are actually people started because the this nutraceutical market is going like instead of exponential, is going like straight. It's very common. People taking a lot of supplements and all there. So people now started looking for drug and the supplements interaction. And there have been cases where some actually nutrients or the supplements actually adversely affect the certain drugs. So doctor advised not to take those particular nutrients when they're taking some drugs. For some drugs, they advise which enhances, for example, omega-3 fatty acids actually potentiate the effects of certain drugs, whereas some drugs that was banned actually, type of Ifidera, I think that for the use for losing body weight, it's been banned because it modifies, it actually affects the cytochrome P450 enzyme directly, and these are the enzymes which involved in drug metabolism. So they reduces the efficacy of the drug. Certainly, there is an interaction, and people need to know. And unfortunately, it's been just started. You know, people start working on it, but I hope to see more data on this in the future.
Any other questions?
Yeah, please.
We already touched upon it, but I was wondering is how does that traditional fuel, how does that traditional food shape the sleeps, and does it also do that in promotion of health, and what happens when you change to another diet?
Well, yeah, I mean, like, you know, it's can, because some of the food involved in the neurotransmission, you know, synthesis, and gene which involved in neurotransmission synthesis, so it can have some emotional or behavior effects too. And then, so what was the first part of your question about?
Traditional food and how changing the diet.
Oh, the diet, yeah. Well, you know, the thing is that you eat traditional food, but not that food, you know, it may be good for you, you don't have to change, but sometimes it may not be good for you. For example, I have lactose intolerance. And then, you know, I was traditionally eating all the dessert and all day, but then I found that, you know, I have lactose intolerance, so I switched. It does not really affect me much because, you know, I get things which are lactose free. So you have to basically, you know, if you learn that some food, you know, even if there's a traditional food but it's not effective, you know, causing problems, then you may have to change. Especially like, you know, lactose intolerance is very common in people in subcontinent. A lot of people, they don't know because it's not lethal and it does not really affect. The only thing happens after taking something with lactose, you get a lot of gas and then you have to go to the restroom. Sometimes, you know, it's very inconvenient because you are walking or traveling or something and you have to go because, you know, and then you have to find a tree. There's no only choice sometimes you get. So this very, you know, not very comfortable situation, but, you know, it's better to know about you and change your diet.
Thank you very much. And now, Professor Gerald can answer this. Professor at the Department of Chemistry, University of Ioannina in Greece. And he is also a adjunct professor at ISES that in Biofinformation. His current interests include NMR spectroscopy as applied to bioorganic chemistry, natural product, analytical chemistry, social biology. He is PhD in 1981 from University of East Anglia in UK. He has 178 papers in different journals. He has been responsible for the university at administrative level, and he included within the best of scientists of their mind discipline. I will ask him to present his work. Very pleased to have him introduced.
Okay. Thank you very much. Ladies and gentlemen, it is a great honor for me to be invited here in this conference in honor of Professor Adel Rahman, since one of his major contributions was the development of high-level postgraduate studies in Pakistan at the level of MPhil and PhD studies. I will not present what I'm doing in Greece. Today, I will simply emphasize what I learned from the published work in international journals of 4 highly talented MPhil students in ICCBs. And I was very lucky to be a co-supervisor for a period of about 3 years. So my presentation will be divided into 4 parts, which are, however, are strongly interrelated. So how to analyze mixtures of lipid hydroperoxides, how to use experimental and computational NMR chemical sets as constraints in order to derive 3-dimensional structures in solution. I will discuss about the structural basis of the so-called eutectic mixtures. And last but not least, I will try to give the molecular basis of aromatic hydrogen deuterium exchange processes. Now, there's no need to emphasize the importance of food lipids. However, cooking, frying, manufacturing, and processing of food may result in oxidation of lipids. Now, this process is extremely complex. And this is due to the fact that the dissociation energies of carbon, hydrogen bonds strongly depends upon the presence of the so-called bis allelic bonds. As a result of that, we have a very significant reduction from 100 kilocal per mole to 65 kilocal per mole. As a result of that, we have the formation of radicals, they interact with oxygen, and we have the formation of hydroperoxides. And those hydroperoxides, depending on the conditions, they can be decomposed into a variety of compounds, aldehydes, alcohols, acids, etc. Now, this process is extremely complex. I mean, if you start just from a single oleic methyl ester, where you have a double bond in cis position, then you end up with a variety of proton NMR chemical sets in this region here. And this is due to the fact that although you start from a cis double bond, you end up with trans bond and also cis. Now the situation is extremely complex because we have the migration of the radical, rotation about double bonds, and in the end, you can have at least final products which are geometric and positional isomers. Rahil decided to tackle this problem using a very simple proton-carbon 13 HNDC experiment. So what he is looking is just a correlation between this proton here and this carbon. And to our surprise, what we realize is that you can have now a clear differentiation between the trans isomers, they are only on a single line with respect to the cis isomers. And this is due to the fact that this carbon here, the chemical state, is extremely sensitive to the geometric isomers, and the chemical state difference is over 6 ppm. So this allows now Rahil to make the complete assignment by using proton-carbon 13 HNDC experiments, although he was looking for minor components in the presence of very, very strong unreacted free fatty acids with resonances with integrals at least 100 times more than the analytes. So this allowed him now to make the complete assignment of proton and carbon 13 chemical sets, even in mixture analysis. But when he tried not to look to polyunsaturated free fatty acids, the situation is very complex. And you might see that in the classical hydroperoxide region around 8 ppm, there are at least 30 different hydroperoxides. And most surprisingly, there were several peaks in the highly distilled region. The original hypothesis was that very probably those are aldehydes, which are secondary oxidation products. But when he added a small amount of PTO, those peaks disappeared. So those peaks are indeed hydroperoxides. But why are those so strongly disabled? The answer is due to the formation of intramolecular hydrogen bonding interactions. But how can we have intramolecular hydrogen bonding interactions? We can have because of the formation of some complex endohydroperoxides. Despite the complexity of those compounds, he was able again to make the complete assignment without any chromatographic isolation. In this case, apart from a two-dimensional proton-carbon 13 NMR spectrum, extremely useful were experiments, one-dimensional toxic experiments. You might see now in the next spectrum, for example, that if you make a selective excitation of this very weak multiplet, then we get a very nice spectrum. Everything else is eliminated. If you do now a selective excitation of this multiplet here, then you get another spectrum. But when he made the analysis of the spectrum, he had it with the same structure, with the same compound. How is it possible to have the same compound and to get completely different proton NMR spectrum? If we look a little bit more carefully on the mechanism, after the reaction with the first molecule of oxygen, we have 1,5 cyclization processes. We have the formation now of a second radical reaction with oxygen, and we end up with the endohydroperoxide, which, however, has 3 asymmetric carbons. So you have the same molecular formula, but then you end up with a total number of 8 stereoisomers, which correspond to 4 pairs of enantiomers. So this is a formidable task in mixture analysis to make the assignment. So we move now to the second part of my presentation. Something which is not more widely appreciated is that NMR chemical shifts are ultimately related to the properties of the bonding electrons around each atom. You do proton NMR, you see everything about protons. You do carbon-13, you see everything about the bonding electrons of the compounds. So the question is it possible to use the experimental chemical shifts, make for various structures computational chemical shifts, and then compare experimental versus computational chemical shifts in order to derive 3-dimensional structures in solution? Can we do that? The answer in principle is yes. We can do density functional theory calculations. And in this case, you can describe a large number of electrons using accurate basis set. But our problem is extremely difficult because you have to differentiate between different diastereomers. But when Rahil made the computations, surprisingly, we realized that different stereoisomers give completely different hydrogen bonding interactions between the hydroperoxide proton with the oxidants of the 5-member ring. So you can make a distinction of the various stereoisomers based on the constraint of the strength of hydrogen bonding interaction. And this is exactly what you can see. Different hydrogen bonding interactions, different chemical states. Of course, this is not the end of the story, because there are also several other hydrogen bonding species, and further research is needed in this direction. Now, in organic chemistry crystallography, incorporation of solvent molecules can be, in most cases, very limited. So, extrapolation of molecular conformations in the crystal to possible conformations in solution, in several cases, may be rather problematic. Simon decided to tackle the following problem. Is it possible to derive structures by incorporating discrete molecules of solvent? So she selected those three compounds. And then she tried now to approach solvent molecules in this region of the spectrum. All her attempts were not successful. This part of the molecule pushes the MSO molecule or water far away, even beyond 4 ounces. But this is not a disappointing result, because in fact, the experimental proton NMR chemical sets of those protons are essentially solvent independent. So we have an agreement now. But this is not the case with this phenol OH group, which shows a chemical set which is strongly dependent upon the solvent which we use. And one can observe chemical state differences up to 5 ppm. Can we use those chemical state criteria in order to define the position of the solvent? She made then a detailed computational work in the gas phase, and she found that indeed we have two conformational states with very little Gibbs free energy difference. In fact, we have the same population. But when she added a solvent molecule, then she derived 3 conformational substates. Of course, one may wonder, what do they mean? Are those really, do they represent what is happening in solution? Or are those 3-dimensional characters? So she decided to do the following. For each of those conformational substates, she calculated the computational proton NMR chemical sets. And since we know computationally which is the Gibbs energy difference between those conformational substates, therefore, she made now an average over the Boltzmann distribution. She has now the computational proton NMR chemical sets, and she decided now to compare with the experimental proton NMR chemical sets. And you can see here that she got an excellent correlation. And then she decided to get the crystal structures, and without any modification, to make calculation of proton NMR chemical sets. And then again to make a comparison with experimental chemical sets. And you see that no correlation. So those computational structures, which are based on experimental proton NMR chemical sets, represent in a much better way the situation in solution rather than X-ray crystallography. Now, motivated by the success of this type of research, we decided to look a little bit on the problem of natural deep eutectic solvents. Everybody knows that sucrose, fructose, glucose, malic acid are solids. But if you take mixtures of them, then they became liquid. And this phenomenon was characterized by one of the leaders in metabolomics, plant metabolomics, Professor Verpute, But this is a simple form of synergy. Very nice word, synergy. This is in fact Greek in origin, but it tells nothing what is going on at the molecular level. Can we do something, because those mixtures are not artificial as might think, because in fact, nectar, which is a liquid, if you take the proton and Mars spectrum, then you can see that it is just a mixture of sucrose, glucose, and fructose. Can we say something about those systems? Now, let's take probably the most characteristic eutectic mixture, DMSO and water. DMSO freezes at 18 degrees Celsius, water at 0. But if you take mixtures, then you can realize that the whole system is completely nonlinear. And there is a very characteristic ratio between water and DMSO, where the freezing point drops to minus 73 degrees Celsius. What is going on here? Sana decided to tackle the problem by using natural products as molecular sensors. And she performed, in fact, some very simple experiments by using this group here, which is extremely sensitive to hydrogen bonding interaction. And she performed some very simple proton NMR experiment by changing the concentration of water. If you do that, then you can immediately see that by increasing the concentration of water, you get a very nice linear correlation. And this is what you would expect. By increasing the concentration in water, you increase the number of hydrogen bonding interactions. But this happens up to a certain maximum, which corresponds to a magic number: 3 molecules of water, 1 molecule of DMSO. After that, although we increase the concentration of water, the chemical shift drops, which means that water at infinite dilution does not provide maximum hydrogen bonding interactions. So we know now the chemical shift in DMSO, we know the chemical shift in water, and we know the chemical shift in this peculiar mixture of water versus DMSO. Can we derive structures? The answer is yes, we can do. And here we have those structures, okay? In water, you can see the arrangement of those 3 molecules of water around the OH group. But with DMSO, you can now immediately, what means synergy? Synergy means now the incorporation of the DMSO molecule of water enhances the hydrogen bonding interaction. And the chemical shift which you can get in both cases agree very well with the experimental proton NMR chemical shift. But even in cases where someone is still very skeptical about the validity of those structures, we were very happy that parallel to the submission of this paper, another paper was submitted using neutron powder diffraction data. So those 2 papers published nearly together. Let's compare now the 2 structures. A DMSO molecule of water, a DMSO molecule of water. First solvation, molecule of water. Second solvation, molecule of water. And surprisingly, the third molecule of water is replaced by the natural product. So we can see now an excellent agreement between the 2 structures. Which is the advantage of this structure? This is a general structure because it tells how natural products interact with the system. You have this cluster here with a maximum hydrogen bonding interactions. The number of hydrogen bonding interactions to the next cluster is significantly reduced, and as a result of that, the melting point reduces significantly. But there is also a very interesting difference between the two structures. For this structure, the scientists used neutron diffraction equipment with an investment of over $100 million. This structure here was produced using just a 400 megahertz instrument and a personal computer. Now, let's move now to the last part of my presentation. Now, it is well known that if we use strongly acidic solutions, then we can get the replacement of an aromatic proton with deuterium through a non-classical sigma carbocation. If we work in basic solution, then we have an exchange through enol keto stoichiometry. Anila decided to investigate this problem, looking on catechin, resorcinol, fluorobus. And to her great surprise, when she performed experiments at neutral pH, then she could observe ready exchange processes for both C6 and C8 protons. This is indeed very surprising. At pH value 7.1. So she was able now to perform different experiments, not only at different pH values, but at different temperatures. By performing very accurate determination of the exchange processes at different temperatures, she was able to define precisely the thermodynamic properties, not of the ground state, but of the transition state. In other words, she was able now to investigate precisely the Gibbs transition energy, the enthalpic transition energy, and the entropic transition energy. I told you before that if you have chemical states in solution, you can produce structures. Of course, in the case of transition state, we cannot do that. Transition states are invisible. However, she suggested that since we know the thermodynamics of the transition state, can we extract the structure of the transition state? Since we know which are the experimental Gibbs energies, which were found surprisingly to be pH independent, contrary to what is in other gravity forces of organic chemistry. Therefore, she started computations. She started from the enol form towards the keto form. But the activation energy was 60 to 55 kilocalories per mole. Therefore, these reaction processes cannot happen. You should wait centuries in order to see a difference in the spectrum. So she decided to do something else, to add solvation molecules of water. 1, 2, 3, 4. And when she added 2 molecules of water, then something extremely important happened. 2 molecules of water in the enol form, 2 molecules of water in the transition state, 2 molecules of water in the keto form. And surprisingly, she got a very, very significant reduction in the activation Gibbs energy, which gives a number of 25 kilocalories per mole, in excellent agreement with the experimental data. I'm not aware of any work in the literature where the transition properties were defined with such an accuracy at the experimental and computational level. Conclusions. Anila suggested that the hydrogen deuterium exchange of aromatic systems is water catalyzed with an acceleration rate of 20 in the 20th power. This is an astronomical number. The second is that acid-basic catalysis accelerates only by a factor of 100, 200, 300. So it's commented that we should change what we teach in undergraduate courses of organic chemistry. And last but not least, in plant metabolomics, when people observe changes and reduction in the amplitude of aromatic signals, they always interpret that in terms of reduction in the concentration of a particular analyte. Anila suggests that one should be very careful, because whenever you use T2O or deuterated methanol, hydrogen-deuterium exchange could play a very significant role. Ladies and gentlemen, it is probably known from the classical years that the Greeks had always a tendency to expand all over the Mediterranean, even in the Black Sea. They created colonies everywhere. But in the south part of Italy and in Sicily, there were so many Greek colonies, so that in Latin language, it was characterized Magna Graecia, Great Greece, because the population there was higher than the population in Sicily and in the city of Syracuse. Archimedes was born, mathematician, physicist, astronomer, and of course philosopher. Let's follow in the YouTube a story about him.
There once was a fellow named Archimedes. He was born in 287 BC in the city of Syracuse in Sicily. He was a Greek mathematician, physicist, engineer, inventor, and astronomer. One day, Archimedes was summoned by the king of Sicily to investigate if he had been cheated by a goldsmith. The king said he had given a goldsmith the exact amount of gold needed to make a crown. However, when the crown was ready, the king suspected that the goldsmith cheated and slipped some silver into the crown, keeping some of the gold for himself. The king asked Archimedes to solve the problem, but there was a catch. He couldn't do any damage to the crown. One day, while taking his bath, Archimedes noticed that the water level in the bathtub rose and overflowed as he immersed himself into the tub. He suddenly realized that how much water was displaced depended on how much of his body was immersed. This discovery excited him so much that he jumped out of the tub and ran through the streets naked, shouting, "Eureka!" which comes from the ancient Greek meaning, "I found it!" What did he find? Well, he found a way to solve the king's problem. You see, Archimedes needed to check the crown's density to see if it was the same as the density of pure gold. Density is a measure of an object's mass divided by its volume. Pure gold is very dense, while silver is less dense. So if there was silver in the crown, it would be less dense than if it were made of pure gold. But no matter what it was made of, the crown would be the same shape, which means the same volume. So if Archimedes could measure the mass of the crown first, and then measure its volume, he could find out how dense it was. But it is not easy to measure a crown's volume. It has an irregular shape that's different from a simple box or ball. You can't measure its sides and multiply like you might for other shapes. The solution, Archimedes realized, was to give the crown a bath. By placing it in water and seeing how much water was displaced, he could measure the volume. Then he'd calculate the density of the crown. If the crown was less dense than pure gold, then the goldsmith most definitely cheated the king. When Archimedes went back to the king and did his test, the story says, he found that the goldsmith had indeed cheated the king and slipped some silver in. These days, using the way an object displaces water to measure volume is called Archimedes Principle. The next time you take a bath, you can see Archimedes Principle in action. And maybe you'll have a genius idea of your own.
Ladies and gentlemen, with all my respect to this historical event, I could attempt and characterize this group of 4 MPhil students as the group of little, very little Eurekas. Although I must confess that I never saw them crying Eureka around ICCBS. Thank you very much.
Thank you so much, Matters. I would like we don't have time to time limit. I would like to thank you for your presentation. I would like also to see that the tea is arranged at Golden Jubilee Hall, kindly processed there for the tea, and the next session of PPS starts sharp at 11.